Right now, if you dug through the rocks beneath your feet and went back far enough, you'd find something deeply strange. For most of Earth's history, almost nothing happened. No fish, no worms, no shells, no eyes, just microscopic blobs of life, doing almost nothing visible for billions of years. 3 billion years, roughly. Then, in what looks like the blink of an eye on the geological timescale, virtually every major animal body plan on Earth appeared at once. Predators, prey, eyes, legs, armor, gills, brains. An explosion of complexity so sudden and so dramatic that for a long time scientists genuinely couldn't explain it. Charles Darwin called it one of the most serious objections that could be raised against his theory of evolution. That's not nothing. That's the guy who invented the theory, admitting it looked broken. So what actually happened? Did animals really appear out of nowhere? Did evolution suddenly slam its foot on the gas after 3 billion years of barely idling? Or is something else going on? Something to do with how rocks preserve life, how oxygen built up in the atmosphere over unimaginable timescales, how a single genetic innovation can unlock an entirely new way of being alive. Those are the questions this video is built around. And before you scroll away thinking this is just another geology lecture, I want to tell you that the answer involves a planetary oxygen catastrophe. A frozen Earth that may have been entirely covered in ice, microscopic cells that essentially invented complex life, and an arms race between predators and prey so intense it rewrote the rules of survival forever. By the end, the Cambrian Explosion won't just make sense. It'll feel almost inevitable, which is somehow even more surprising. Here's the central paradox we're going to unpack. When geologists look at rocks from around 540 million years ago, the fossil record goes from nearly empty to extraordinarily full within what appears to be a geologically short window. Shells, skeletons, eyes, complex bodies, all showing up fast. But fast in geology can mean millions of years. So the real question isn't just what caused this burst of life. It's whether it was actually sudden at all, or whether we're looking at an illusion created by the limits of fossilization, the chemistry of ancient oceans, and gaps in the rock record that swallow entire chapters of evolutionary history whole. We'll get into all of it. The oxygen story, the genetics, the ecology, the fossils that either don't preserve or simply weren't there yet, and how scientists use chemistry baked into ancient stone to read time periods where rocks themselves barely survived. If you're listening to this while falling asleep, that's honestly fine. That's part of why I make these. Drop a comment and tell me where you are and what time it is for you. I check every one of them, and every time it's someone in Tokyo at midnight and someone in Toronto at 7 a.m., and somehow that never gets old. Now, let's go back to the beginning. The actual beginning. The actual beginning is not a good place for life. Picture Earth around 4.5 billion years ago. The planet is essentially a molten rock being pounded constantly by asteroids, volcanic eruptions covering entire continents, a sky full of toxic gases, and no ozone layer to stop radiation from sterilizing anything that dares to exist on the surface. The oceans, once they form, are not blue. They're probably a kind of rusty greenish murk, hot, acidic, and almost entirely devoid of free oxygen. We call this the Hadean, named after Hades, which tells you everything you need to know about what early Earth thought of hospitality. And yet, somewhere in that hostile chemical chaos, something started. The earliest evidence we have for life on Earth pushes back to around 3.7 to 4 billion years ago, though the oldest claims are still debated. What we're talking about isn't animals, isn't even cells with nuclei. We're talking about bacteria and archaea, microscopic single-celled organisms so simple they don't even have a membrane-wrapped nucleus to hold their DNA. Just free-floating genetic material in a bag of chemistry, reproducing, mutating, dying, and doing it all over again. For approximately 3 billion years, that was basically it. Now here's where the title of this video needs a serious correction, because the phrase "nothing evolved" is wildly, almost offensively wrong. Devolution never stopped, not for a single generation. What happened during those 3 billion years was relentless, constant, ruthlessly efficient evolutionary change. It just happened to be invisible. Not invisible because nothing was happening, but invisible because everything happening was microscopic, mostly soft-bodied, and radically difficult to preserve in the fossil record. Think about that carefully. 3 billion years of evolution, and we have almost nothing to show for it in terms of obvious fossils. The question isn't why evolution stalled. The question is why we can barely see it. Part of the answer is preservational. Fossilization is not a democratic process. Hard shells, bones, teeth, the mineralized structures that make up most of what we think of as fossils, those preserve beautifully under the right conditions. But bacteria don't have any of that. A single bacterial cell is roughly 1 micrometer across. Soft, chemical, easily dissolved by the same geological processes that crush and heat and transform rock over millions of years. Even when microbial communities do leave traces, identifying them with certainty is genuinely hard. There are structures that might be ancient microbial fossils, and there are geochemical formations that look exactly like ancient microbial fossils but aren't. The line between the two has caused decades of scientific arguments. There is, however, one exception: stromatolites. Stromatolites are layered rock structures, essentially fossilized microbial mats, built up over time by communities of cyanobacteria and other microbes that trap and bind sediment as they grow. They look like lumpy mushrooms or layered cabbages made of stone, and they show up in the fossil record as far back as 3.5 billion years ago, possibly further. For an enormous stretch of Earth's history, stromatolites weren't just present, they were the dominant visible ecosystem on the planet. Ancient shallow seas were lined with them. They were the coral reefs of their era, except instead of supporting thousands of animal species, they were primarily built by and for microbes. Here's what makes stromatolites genuinely important rather than just geologically interesting. The organisms building them, particularly cyanobacteria, were doing something with enormous consequences. They were performing oxygenic photosynthesis, splitting water molecules using sunlight and releasing oxygen as a byproduct. Every single oxygen molecule you're breathing right now traces back to that microbial process, started by organisms that looked like green slime and lived in flat mats on the seafloor billions of years ago. They were quietly, invisibly, re-engineering the entire planetary atmosphere. We'll get into that in the next section because it's its own story. But the point right now is that the microbial world wasn't static. It was transforming Earth on a planetary scale. It just wasn't doing anything that looked dramatic when you were standing on the shore. That gap between significance and visibility is one of the central ideas you need to hold on to throughout this entire story. Because here's something even stranger to consider. Some researchers think the 3 billion year microbial era wasn't just a waiting room for animals to eventually show up. It may have been a period of profound, cryptic biological innovation. Innovations that left almost no obvious fossil signature, but fundamentally reorganized the rules of life on Earth. The development of new metabolic pathways, the invention of different ways to handle energy, oxygen, sulfur, iron, the gradual evolution of more sophisticated cell-to-cell communication in microbial mats. These changes don't leave the kind of evidence that makes it onto a museum placard, but they may have been setting biological and chemical preconditions for everything that came later. Think of it like the infrastructure work that happens before a city gets built. Nobody makes documentaries about the people laying sewage pipes and running electrical cables, but without those invisible foundations, nothing else can exist. The microbial era was laying pipe for 3 billion years. By the time we reached the mid-Proterozoic, roughly 1.5 to 1.8 billion years ago, something genuinely new had appeared. Cells with nuclei, eukaryotes, organisms dramatically more complex than anything that had come before, capable of storing more genetic information, dividing in more sophisticated ways, and eventually doing something that single-celled life had never really managed on a large scale. Building bodies made of multiple different types of cells. That's coming. But the point for right now is that even this revolution, the invention of the kind of cell that every animal on Earth is built from, happened deep in the Proterozoic and left a fossil record that's fragmentary, ambiguous, and often nearly invisible. We know it happened largely because the descendants of those cells are alive today. The evidence was written not in stone, but in the molecular machinery of every living thing on the planet. 3 billion years of life, constant evolution, planetary-scale consequences, and almost nothing to show for it in a museum case. That's not evolution failing. That's evolution doing exactly what it does, which is change what it can, with what it has, slowly and invisibly, and without any particular regard for whether we'll be able to find the receipts later. So let's talk about oxygen. Because if there's one molecule that keeps appearing in every serious discussion of why animals eventually evolved, it's this one. But the story is not clean, and it is not simple. And anyone who tells you oxygen caused the Cambrian Explosion in a straight line from A to B is skipping over about 2 billion years of deeply inconvenient complications. Start with the basics. Photosynthesis, the process those cyanobacteria in the previous section were running, splits water molecules using sunlight and releases oxygen as a waste product. The cyanobacteria weren't trying to oxygenate the planet. They were just eating light and dumping what they didn't need. But they were doing it in enormous numbers, in shallow seas across the entire planet, for hundreds of millions of years, and the oxygen piled up, slowly, grudgingly, in fits and starts that took geological timescales to register. Here's the thing about early Earth that most people don't appreciate. The planet was not waiting around to receive oxygen. It was actively consuming it. The ancient oceans were full of dissolved iron, pumped out by volcanic vents on the seafloor in extraordinary quantities. And iron, when it meets oxygen, does exactly what a piece of iron left in the rain does. It rusts. That dissolved iron grabbed the oxygen being produced by cyanobacteria almost as fast as it was released, pulling it out of the water and dumping it on the seafloor as iron oxide minerals. Those minerals are still there. We call them banded iron formations, and they're essentially a geological record of oxygen being produced and immediately consumed, layer by layer, for hundreds of millions of years. The planet had a sink, a massive, nearly bottomless chemical drain that swallowed oxygen before it could accumulate anywhere meaningful. Add to that volcanic gases. Early Earth was volcanically hyperactive, and volcanic gases like hydrogen sulfide and sulfur dioxide react with oxygen and remove it from circulation. The atmosphere and oceans had enormous chemical buffers working against any net oxygen accumulation. The cyanobacteria were essentially filling a bathtub with the drain wide open. Then, around 2.4 billion years ago, something shifted. We call it the Great Oxidation Event, and the name sounds dramatic enough to suggest animals should have appeared immediately afterward. They did not for another 2 billion years. What actually happened was that the iron sinks in the oceans gradually became saturated. The volcanic outgassing rates may have declined somewhat, and the cyanobacteria kept producing. Eventually, the production rate outpaced the consumption rate, and for the first time in Earth's history, free oxygen began accumulating in the atmosphere. Not a lot by modern standards. Early estimates suggest atmospheric oxygen reached somewhere between 1% and 10% of present levels. But it was a permanent change, a threshold crossed. The planet's chemistry would never be the same, and yet nothing with a backbone, nothing with a mouth, nothing recognizable as an animal appeared for roughly 2 billion more years, which should give you serious pause if you're tempted to draw a straight arrow from oxygen to animals. The reason scientists know any of this, by the way, is not because they were there. It's because certain elements behave differently depending on how much oxygen is present in the environment where they're deposited. Molybdenum and uranium, for example, are what geochemists call redox-sensitive metals. In low oxygen conditions, they stay locked in rock. In oxygenated water, they dissolve and get carried into sediments. By measuring concentrations of these metals in ancient rock layers, scientists can build a rough timeline of when oxygen was present, where, and in roughly what quantities. They also use sulfur isotopes, carbon isotopes, and the presence or absence of certain mineral phases that only form under specific redox conditions. But here's the honest caveat. These proxies measure local and regional conditions in the specific environments where those rocks were deposited. Stitching that data into a global picture involves significant assumptions, and different research groups looking at the same data sometimes reach different conclusions about the precise timing and magnitude of ancient oxygenation events. The broad strokes are solid. The fine details are actively argued over. What seems increasingly clear is that oxygen did not rise smoothly. It rose in steps. A significant jump at 2.4 billion years ago, then a long plateau, sometimes called the boring billion, where levels may have actually stayed relatively low and stable for an enormously long stretch of time. Then, intriguingly, another pulse of oxygenation much later, in the Neoproterozoic, somewhere between roughly 800 and 540 million years ago. And that second pulse lines up much more closely in time with the appearance of animals. This is where oxygen becomes genuinely interesting as a potential factor rather than a distant background condition. Animals, unlike bacteria, are metabolically expensive. Moving a body through water, building complex tissues, running a nervous system, hunting prey. These are energetically demanding activities that require sustained aerobic metabolism at levels that simply cannot be supported at very low oxygen concentrations. There's a rough threshold below which you cannot build and power a large, active, mobile organism. The exact number is debated, but the principle is solid. Bigger, more complex, more active animals need more oxygen. So when geochemical proxies suggest that deep, ocean waters, which had likely remained anoxic long after the surface oxygenated, may have finally begun receiving more oxygen during the late Neoproterozoic, that's not a trivial detail. That's potentially the last lock coming open on a door that had been sealed for billions of years. But oxygen alone almost certainly wasn't enough. It may have been necessary without being sufficient, because plenty of things had to be invented before any organism could use that oxygen to build a body worth talking about. And that invention, perhaps the most consequential in all of evolutionary history, happened at the cellular level, long before the first animal drew its first breath. To understand why animals took so long to appear, you have to zoom in, way in, past continents and oceans, past ecosystems, down to the level where organism still just means a single cell trying not to die. Because before you can have a fish or a worm or a trilobite, you need a cell that can even imagine being part of something that big. And for the first half of Earth's history, that cell did not exist. For billions of years, life was almost entirely prokaryotic. Bacteria and archaea, tiny, tough, astonishingly versatile, but simple in one crucial way. No internal compartments, no nucleus, no mitochondria, no little organelles doing specialized jobs. Just a membrane, a loop of DNA, some ribosomes, and chemistry. Prokaryotes are masters of efficiency. They're small because they have to be. Everything they do, energy generation, DNA replication, protein synthesis, happens in the same cramped space. That works brilliantly if you're a microbe dividing every 20 minutes. It does not scale if you want to build a cell big enough to house a complex genome, let alone coordinate with millions of other cells in a body. Eukaryotes are the opposite strategy. These are the cells all animals, plants, fungi, and a lot of weird microbial lineages are built from. They have a nucleus to store and protect their DNA. They have an internal skeleton made of protein filaments that lets them change shape, engulf food, move around. They have membrane-bound organelles that specialize just like organs in a body. On paper, this sounds like a minor upgrade. In practice, it's a categorical shift. Prokaryotes are like a one-room cabin. Eukaryotes are like a factory complex with separate buildings, power lines, and a management office. You can do things in a factory you will never do in a cabin, no matter how long you sweep the floor. The big question is how that factory got built. Somewhere more than 1.5 billion years ago, maybe earlier, an ancestor of modern eukaryotes did something microbes had probably been doing for a long time. It swallowed another cell. Many bacteria live inside other cells as parasites or prey. Usually the story ends the same way. One of them dies and gets digested. But this time, it didn't. The leading idea, endosymbiosis, says that an archaeal cell engulfed a bacterium, and then, instead of killing it, made a deal. The host provided protection and nutrients. The bacterium paid rent in ATP, the chemical energy currency of all living cells. Over time, that once free-living bacterium shrank, lost most of its genes, and became what we now call a mitochondrion. This is not a fringe theory. You can still see the evidence inside your own cells. Mitochondria have their own tiny circular genomes, more like bacterial chromosomes than anything in a eukaryotic nucleus. They divide independently of the cell they're in using a bacteria-style process. Their membranes and some of their proteins look distinctly bacterial. It's as if evolution left the receipts. Why does this matter for animals? Energy. Running a large genome, proofreading it, transcribing it, translating it into proteins, regulating those proteins in space and time, that is energetically expensive. A prokaryote generates energy across its outer membrane. Try to scale that up, and you hit a geometry problem. Volume grows faster than surface area. You simply can't supply a big cell with enough power through one membrane. Mitochondria solve that by bringing the power plants inside. Each one has a highly folded inner membrane packed with the machinery of respiration. A single eukaryotic cell can have hundreds or thousands of these internal power stations. More membranes, more energy, without needing to enlarge the cell's outer surface. Some researchers argue that this was the key move that allowed cells to carry much larger genomes and run much more complex regulatory networks. In that view, endosymbiosis wasn't just an interesting side story. It was the energy upgrade that made complex life physically possible. But energy and complexity at the single-cell level still don't get you animals. A giant amoeba is not a jellyfish. To build bodies, you need something else: cooperation. Multicellularity sounds like a binary condition. You either are or you aren't. In reality, it's more like a spectrum that life wandered onto again and again. There are green algae that normally live as single cells but sometimes clump together into simple colonies. There are slime molds that spend most of their time as solitary amoebas, then merge into a slug-like mess when food runs out. Some cells sacrificing themselves to help others disperse. Fungi, brown algae, red algae, land plants, animals. All of them represent separate experiments with being multicellular. This tells you something important. There was no single magic moment when multicellularity appeared, and the universe applauded. It was a theme evolution kept returning to, refining it, elaborating it, solving the same engineering problems in different ways. Those problems are basic but brutal. If you're going to be more than one cell, your cells have to stick to each other. They have to talk to each other, and they have to agree on who does what and when. Modern animals handle sticking using an entire toolkit of adhesion proteins, cadarins, integrins, and others that function like molecular Velcro and bolts. These proteins let cells form tissues, maintain boundaries, heal wounds. They're absolutely fundamental to being an animal. The weird thing is, the genes for many of these adhesion molecules show up in single-celled relatives of animals, chionoflagellates, for example, organisms that never form a nervous system, never grow a muscle, but still seem to have a starter kit for animal-style cell biology. Same with signaling. Animal cells talk to each other using secreted molecules and surface receptors. They coordinate development, immune responses, tissue maintenance. But when biologists sequenced the genomes of close unicellular cousins to animals, they kept finding pieces of those same signaling pathways already present, doing simpler jobs. Even the core tools that control when genes turn on and off, the transcription factors and regulatory switches that pattern an embryo, have deep roots in pre-animal lineages. The implication is that the genetic and cellular toolkit for building a body did not appear overnight with the first animal fossils. It was assembled gradually inside single cells that were never animals at all. The rocks give us a hazy glimpse of this transition. The earliest widely discussed eukaryote fossils are microfossils, tiny, complex-walled cells preserved in ancient shales and cherts. Some date back to at least 1.6 to 1.8 billion years ago, maybe even older depending on which interpretation you favor. They have features like thick outer walls, spines, or internal structures that look more like eukaryotes than bacteria. We also have molecular fossils, biomarkers. Certain organic molecules, like sterains derived from sterols, are strongly associated with eukaryotic cell membranes. Their appearance in sedimentary rocks has been taken as evidence that eukaryotes were around and doing business even when body fossils are scarce or ambiguous. But evidence here comes with big caveats. Ancient rocks get heated, squashed, infiltrated by fluids. Modern contaminants can sneak into old samples. Some biomarkers once thought to be unambiguous eukaryotic signatures have been reinterpreted or challenged. Even the identity of some microfossils is actively argued. Are we looking at true eukaryotic algae or oversized bacteria with fancy cell walls? So the timeline is fuzzy. What seems likely, though, is that eukaryotes were present and diversifying hundreds of millions of years before animals show up in any obvious way. The cellular revolution quietly preceded the visible revolution. Somewhere in that long, murky interval, eukaryotes didn't just get mitochondria and nuclei. They got increasingly sophisticated internal skeletons that allowed them to change shape, engulf prey, and divide labor inside cells. They evolved adhesion molecules that started as tools for sticking to surfaces or capturing food, but later became the glue between neighboring cells. They evolved signaling pathways that began as environmental sensors and eventually became intercellular communication channels. Piece by piece, the problem of building bodies was being solved in advance. Long before there were worms burrowing in the seafloor, there were single cells experimenting with the molecular language that would one day coordinate trillions of cells in an animal. And all of this was happening under shifting environmental conditions shaped by oxygen. Rising oxygen made high-energy lifestyles possible. Eukaryotic cells, with their internal power plants, could exploit that. Then, as these cells learned to stick together and talk to each other, they laid the groundwork for something entirely new. Organisms whose size and complexity weren't limited by a single cell's geometry. By the time oxygen in the oceans and atmosphere finally approached levels that could sustain big, active bodies, the universe wasn't starting from scratch. It had a toolkit ready: eukaryotic cells with mitochondria, adhesion, signaling, and embryonic versions of developmental control systems. The stage was set, the plumbing was in, the power grid was online. What remained was to stress test this new infrastructure under the most extreme conditions the planet had experienced in over 1 billion years. That test came in the form of ice, possibly a whole planet's worth of it. Imagine waking up in a world where the horizon in every direction is white. No waves, no open ocean. Just a solid, grinding shell of ice, maybe a kilometer thick in places, stretching from pole to pole. Temperatures far below freezing, sunlight bouncing off a global mirror. This is the extreme version of an idea geologists call snowball Earth. The basic claim is simple but brutal. Several times between about 720 and 635 million years ago, during what's called the Neoproterozoic, Earth might have frozen over almost completely. Oceans capped in ice, continents buried under glaciers that reached nearly to the equator, a planet that looks less like blue marble and more like a white cue ball. That sounds insane. So why do people take it seriously? Because the rocks, inconveniently, keep hinting at something like this. On multiple continents, right around the same ages, you find layers of rock that can only really form in glacial environments. Tillites, dropped boulders, striated surfaces that look like they've been scraped by ice. Nothing weird there if they were near the poles. But many of these deposits sit on rocks that formed in the tropics. You can tell from the way Earth's ancient magnetic field is recorded in them. That suggests glaciers, or at least big ice sheets, reached all the way into what should have been warm, sunny latitudes. On top of those glacial layers, almost like frosting on the ice cake, you often find another strange signature: cap carbonates. These are thick, rapidly deposited layers of carbonate rock, limestones and dolostone, that seem to have formed in warm, shallow seas. They're loaded with chemical and isotopic clues that point to intense weathering and wild changes in the carbon cycle. Glaciers at the equator, then a sudden switch to hot, carbonate-precipitating oceans, over and over again at roughly the same times globally. It looks suspiciously like the world flipped between two climate extremes. That's the core snowball Earth story. Planet freezes, then planet falls violently. But as always, the details are messy, and not everyone buys the full, frozen cue ball picture. Some researchers argue for a slush ball Earth instead. In that version, ice spreads far into the tropics, but not everywhere. Maybe belts of thin, seasonally open water hug the equator. Maybe some pockets of ocean near volcanoes or upwelling currents stay partly ice-free. That would matter a lot for life, because completely sealing the oceans under thick ice is a very different biological problem than leaving a few liquid refuges. Either way, nobody thinks the climate was calm and pleasant. The Neoproterozoic looks like a time when Earth's thermostat went haywire. Multiple major glaciations, rapid swings, and long intervals where the planet was locked into a deeply icy state. And if you're trying to understand why animals appear shortly after this period, you need to ask, what does a planet-scale freeze do to life, and to the chemistry life depends on? Start with nutrients. Under normal conditions, continents weather. Rain, slightly acidic from dissolved CO2, breaks down rocks. That process releases ions, calcium, magnesium, phosphorus, iron, silica, into rivers, which carry them to the sea. There, they feed plankton and other microbes, which then both drive and respond to global cycles of carbon, nitrogen, and oxygen. Cover everything in ice, and you choke that system. Glaciers grind rock into fine powder, but if the planet is mostly frozen, very little liquid water is around to transport those nutrients into the ocean in a steady way. Photosynthesis near the surface could be throttled. Light has to fight its way through meters of ice and snow. Circulation patterns in the ocean change when you put a rigid lid on top. Oxygen levels in deeper waters may drop if less organic matter is sinking, and fewer currents are mixing the water column. Life doesn't necessarily disappear under those conditions, but it's pushed into tight corners. Microbes hugging hydrothermal vents, bacterial mats beneath thin ice, in rare open water leads or coastal refuges, organisms in subglacial lakes, living in dark chemistries. This is where the analogy to a bottleneck comes back. Just like that tiny population of early humans clinging to survival in scattered refuges, Neoproterozoic life may have been squeezed into microhabitats where only certain strategies worked: slow-growing, tough, stress-tolerant, able to handle low nutrients and low light. On a planet scale, that's an ecological bottleneck. A lot of diversity may have vanished. Many lineages that had been quietly experimenting with new ways of being eukaryotic for hundreds of millions of years could have been pruned away. The fossil record from these intervals is sparse and controversial, so we don't have a clear casualty list. But extreme, long-lasting glaciations are not gentle filters, and yet the aftermath of a snowball looks like a setup for radical change. Think about how you get out of a global ice state. Once you've covered the world in reflective ice, sunlight mostly bounces back into space. Earth cools, which grows more ice, which reflects more light. A positive feedback loop that tends to lock the planet in a frozen condition. The way out in most models is volcanoes. Volcanoes keep erupting even when the surface is frozen. They keep belching CO2 and other gases into the atmosphere. Under normal warm conditions, that CO2 would help drive weathering, and weathering would pull it back out of the air, locking it into rocks. But if the continents are covered in ice, that feedback is mostly shut off. CO2 builds up, slowly, relentlessly. Over millions of years, atmospheric CO2 could climb to levels tens or even hundreds of times higher than today's. At some point, the greenhouse effect overwhelms the ice albedo effect. The planet starts to warm. The first patches of ice start to melt. Dark rock and liquid water appear, absorbing more sunlight. Warming accelerates. The ice retreats in a geologic flash. This is where things get chemically wild. You've just exposed continents that have been sitting under glaciers for millions of years. Those glaciers have pulverized rock into fine reactive powder. Now suddenly, warm rain falls on this stuff in an atmosphere loaded with CO2. Weathering goes into overdrive. All those ions, calcium, magnesium, phosphorus, iron, rush into rivers and then into the oceans. The seas, which may have been relatively stratified and nutrient-poor in many places during the deep freeze, suddenly get a gigantic fertilizer dump. Cap carbonates record one part of that story. Rapid precipitation of carbonates is the ocean. An atmosphere adjusts to this pulse of chemistry. But for life, the key is that you've taken a starved system and turned on the food tap. In many models, post-snowball oceans become nutrient-rich, especially in phosphorus and some trace metals that limit productivity. Microbes that can photosynthesize, algae, cyanobacteria, other plankton, respond by blooming like crazy. Organic carbon production skyrockets. Oxygen production, tied directly to this photosynthesis, ramps up as well. This could help explain geochemical hints that, after these Neoproterozoic glaciations, oxygen levels in the oceans and atmosphere stepped up, at least regionally and intermittently, toward values that could support big, active, multicellular organisms. So you have this sequence: long cold squeeze, then hot, nutrient and CO2-rich rebound. From an evolutionary perspective, that's not just a catastrophe and a recovery. It's a reorganization. During the glacial phases, lineages that can handle extremes are favored. Things that can insist, slow their metabolism, tolerate anoxia or high salinity, form biofilms, exploit chemical energy deep in the crust. Those get through. Many others don't. When the ice retreats and nutrients flood in, the survivors find themselves in a world full of open ecological space. Vacant niches, underutilized energy sources, new gradients of light, temperature, chemistry. That's a classic recipe for evolutionary experimentation. You can think of it as pressing a reset button on ecosystems. Old equilibria are gone, food webs are thin, competitive landscapes are scrambled. In that context, traits that were marginal or expensive before can suddenly become winning strategies. Maybe some eukaryotic lineages that had dabbled in simple multicellularity, forming filaments, sheets, or small colonies, discover that in a nutrient-rich, oxygenated post-glacial sea, being bigger helps you stay in the light or avoid being eaten. Adhesion molecules and signaling pathways that previously just helped single cells stick to surfaces now become tools for forming more persistent bodies. Maybe predation pressures shift. New microbial predators and grazers appear, selecting for defenses, including larger size and more coordinated behavior. Maybe patchy oxygen creates vertical habitats, oxyclines in the water column and sediments, where multicellular organization helps a lineage exploit multiple layers at once. The important point is not that Snowball Earth directly caused animals. That would be oversimplifying. The timing is suggestive. Complex multicellular fossils and the first macroscopic eukaryotes start appearing in the Ediacaran, soon after the last of the Neoproterozoic glaciations. But many other factors were in play: tectonics, nutrient cycles, pre-existing genetic toolkits. What global glaciations likely did was shake the table. They erased a lot of incumbents. They rearranged energy flows between the atmosphere, ocean, and biosphere. They altered the availability of key nutrients and possibly pushed oxygen levels higher in pulses. In doing so, they created both bottlenecks, harsh filters that few lineages passed through, and bursts of opportunity for those that did. And this didn't happen just once. The Neoproterozoic hosts at least two major, possibly global glaciations, the Sturtian and Marinoan events, and probably several less extreme but still significant ice ages. Each may have played out differently, different continents in different positions, different ocean gateways, different pre-existing ecosystems. The rock record is patchy. Some basins preserve beautiful sequences of glacial deposits, cap carbonates, and post-glacial sediments. Others have gaps, metamorphism, or later tectonic overprints that blur the details. Dating these events precisely is hard. Reconstructing exactly how far the ice spread, how long it lasted, and what conditions were like under the ice is harder still. So when people talk about Snowball Earth, they're really talking about a family of scenarios: full global freeze versus slush ball, thin equatorial refugees versus thicker belts of open water, shallow seas that flip from anoxic to oxygenated versus deeper basins that lag behind. For evolution, those differences matter. A world with scattered open oceans offers more continuous habitat than a truly sealed snowball. That might mean less severe bottlenecks, different styles of survival, different starting conditions when the ice retreats. What almost everyone agrees on, though, is that the Neoproterozoic was not a boring prelude. It was a time of radical climate swings, tectonic rearrangements, and big shifts in ocean chemistry. A time when the environment refused to sit still, and life, which had spent billions of years refining single cells and simple colonies, suddenly had to prove just how adaptable those new eukaryotic toolkits really were. Out of that long freeze-thaw drama, something new began to appear in the rocks. The first large, soft-bodied organisms. Not yet trilobites or clams or anything you'd recognize from a marine field guide, but genuine macroscopic life decorating the seafloor. The Ediacaran world was about to unfold, a strange, alien ecosystem built on the ruins and opportunities Snowball Earth left behind. Picture wading out onto a shallow Neoproterozoic seafloor just after the last great ice retreats. The water is still, a little murky with fine sediments washed off freshly exposed continents. Sunlight filters down for a few meters of water. There are no fish, no crabs, no swaying kelp forests, no shells crunching underfoot. What you see instead is a kind of alien garden. Flat quilted ovals lie like throw pillows on the mud. Frond-like structures rise from the seafloor, some as tall as you are, branching into delicate leaf patterns and gently swaying in the current. Circular discs, rings, and ridged spirals are stamped into the sediment, like someone's been pressing cookie cutters into the ocean floor. This is the Ediacaran world, roughly 575 to 541 million years ago. It's the bridge between 3 billion years of mostly microscopic life and the animal-packed oceans of the Cambrian. It looks peaceful. It was also deeply strange. The organisms we find from this time are so odd that for over a century paleontologists have argued about what they even are. Take the fronds. In places like the famous Mistaken Point in Newfoundland or the Ediacara Hills in Australia, you see whole bedding plains covered in fossil forests of things like Charnia and Charniodiscus. They look like ferns or sea pens, a central stalk, with side branches arranged in repeating patterns, sometimes in a graceful fractal, where each little branch looks like a miniature version of the whole, but they're preserved lying flat in volcanic ash or fine sediment, like plants pressed in a book. No roots, no obvious mouths, guts, or eyes, no shells, just soft leaf-like bodies. Then there are the classic quilted forms. Ranger, Dickinsonia, Sprigina, and their kin look like someone cut out shapes from a padded comforter and dropped them on the seafloor. Bilateral ovals, teardrops, and ribbons divided into repeated pillow-like segments. Sometimes those segments branch off at angles in a left-right pattern that looks almost like ribs. These things can be big. Some Dickinsonia reach over 1 meter long. For most of Earth history up to this point, you'd have needed a microscope to see life. Suddenly you have mattress-sized blobs lounging on the ocean floor, and then the discs, Aspidella, Tribrachidium, and others show up as circles, 3-armed pinwheel patterns, concentric rings, sometimes with radial grooves. They might be attachment discs for fronds. They might be independent organisms. They might be something else entirely. If this all sounds hard to picture clearly, that's sort of the point. The Ediacaran biota doesn't map cleanly onto anything alive today. That's where the arguments start. One camp says, these are early animals, maybe weird, maybe soft-bodied, but on the same major branches of the tree of life that later gave us jellyfish, worms, and eventually vertebrates. There's some evidence for that. A few Ediacaran forms, like Cambrian, look suspiciously like bilaterally symmetrical animals, with a clear front and back, left and right. Their fossils are found alongside scratch marks in the sediment that look like grazing trails. That suggests a creature with a muscular foot or belly, slowly moving across microbial mats and scraping food as it went. Some Dickinsonia fossils show hints of internal complexity, and a growth pattern that looks more like animals than fungi or algae. Recently, researchers claim to find traces of cholesterol-like molecules in Dickinsonia fossils, steroids often associated with animals. Not everyone is convinced, but it's one more piece of the puzzle. Another camp says, these are almost all failed experiments. On this view, Ediacaran organisms belong to a largely extinct group, or groups, of multicellular life that tried out large body size and complex shapes, colonized seafloors worldwide for tens of millions of years, and then vanished without leaving direct descendants. The best-known version of this idea calls many of them Vendebiants, a hypothetical kingdom of fractal, quilted organisms that aren't quite animals, plants, or fungi. Their quilted anatomy looks efficient for distributing nutrients across a large surface without internal plumbing. Their growth patterns, in some analyses, don't match those of modern animals. They may have fed in ways that are rare or absent today. A third angle goes even further. Maybe some of these fossils aren't bodies in the normal sense at all. They might be large, complex structures built by microbial communities, thick biofilms and mats that grew, buckled, and textured the sediment. Or they might be organisms tightly tied to those mats, more like elaborately organized microbial skins than independent animals crawling around. So which is it? Early animals, extinct experiments, or microbial constructions? The honest answer so far is all three might be in the mix. The Ediacaran fossil record is a grab bag. Lumping everything into one category has not worked well. As more fossils are found and more detailed analyses are done, a picture is emerging where some forms really do seem close to the base of animal lineages. Some are probably total evolutionary dead ends, and some may be complex microbial life wearing an animal-shaped disguise. To make sense of this ecosystem, you have to look beyond body fossils and pay attention to something more subtle. Trace fossils. Body fossils are the actual remains or impressions of organisms. Trace fossils are the marks they leave behind. Burrows, trails, feeding scratches, resting impressions. In late Ediacaran rocks, you start to see faint, meandering trails on and just below the sediment surface. Simple, shallow, often only a few millimeters wide. They wiggle, turn, cross themselves, and sometimes radiate out from a central point like the tracks of something exploring. These traces are usually attributed to small, soft-bodied animals, worm-like creatures or slug-like grazers, moving through or across the microbial mats that carpeted the seafloor. Even when we don't see their bodies preserved, their behavior is written into the sediment. Some traces suggest a kind of decision-making, turning when encountering obstacles, spiraling as if searching for food, stopping and starting. That implies nervous systems, muscles, front ends that sense the environment, and back ends that follow. It's hard to overstate how revolutionary that is. For billions of years, the seafloor was dominated by mats of microbes, cyanobacteria, algae, and other single-celled organisms forming smooth, rubbery carpets. In the Ediacaran, those mats are still there, but now something is moving across them, into them, under them. The mats themselves shape almost everything about Ediacaran life. Many large Ediacaran organisms seem to simply lie on top of them, like solar panels on a roof, maximizing contact with the chemical-rich, oxygenated interface between water and sediment. Some may have anchored into them with holdfast discs like Charnia and its kin, raising their fronds into better-flowing water to capture dissolved nutrients or tiny particles. Others, especially the quilted mattresses, might have fed by osmotrophy, absorbing dissolved organic matter directly across their large surfaces, using the microbial mat's constant X states as a buffet. In that scenario, being big and flat is an advantage, not for hunting but for soaking. The grazers and burrowers in turn are exploiting the mats in a more active way, scraping, nibbling, disturbing. Their traces show where they systematically mowed patches of microbial carpet before moving on. Movement is mostly gentle, gliding, creeping, inching through the top few millimeters of sediment. No deep burrows yet, no signs of major disturbance. The seafloor in many places is still a relatively undisturbed blanket, decorated rather than demolished by life. It's a world where ecosystem engineering is just beginning. You have sessile fronds filtering or absorbing, big flat osmotrophs basking on microbial lawns, small mobile grazers leaving calligraphy-like tracks, and perhaps a few early predators or scavengers nibbling at soft tissues. Predation, as far as we can tell, is minimal. Armor is rare. Skeletons are almost non-existent. Tissues are soft. Many organisms are simply pasted to the substrate, relying on being inedible, tough, or chemically defended rather than armored or mobile. That tranquility doesn't last. By the very end of the Ediacaran, something changes in the trace fossil record. You start to see more complex burrows, deeper penetration into the sediment, more intense disruption of microbial mats. A style of living inside the seafloor rather than just on top of it begins to take hold. At the same time, the classic quilted and frondose Ediacaran forms begin to decline. In some regions, they vanish abruptly right at the boundary with the Cambrian. In others, they fade out over several million years. Either way, the distinctive Ediacaran gardens give way to more mixed communities. Small shelly fossils, mobile bilaterians, worm tubes, and other things that look more obviously animal. Why? There's no single agreed-on answer, but several pressures line up suspiciously well. One is the rise of active burrowing. Once worms and other early animals start churning up the sediment, they literally break the foundation that many Ediacaran organisms depended on. Microbial mats get shredded, oxygen penetrates deeper, chemical gradients shift. A whole style of life built on stable laminated carpets gets undermined. Another is increasing predation and grazing intensity. As more mobile, muscular animals evolve, soft, exposed, slow-growing quilted forms become easy targets. Even a modest increase in tearing, rasping, or biting pressure could push them into decline if they can't evolve defenses fast enough. Environmental changes may also have played a role. Fluctuating oxygen levels, shifting nutrient regimes, and changing sedimentation patterns as continents moved and erosion patterns evolved. The Ediacaran world was not static. It was part of an ongoing Neo-Proterozoic roller coaster. The result is tension that sets the stage for the Cambrian. On one side, you have the long-established mat-based Ediacaran ecosystem. Large, soft, mostly peaceful, feeding gently off chemical fluxes at the seafloor. On the other, you have the early stirrings of a more disruptive, animal-dominated world. Burrowers, grazers, possibly early predators, testing new ways to move, eat, and interact. For tens of millions of years, these two modes of life overlap and interact. But as the Cambrian approaches, the balance starts to tip. The weird garden of quilts and fronds begins to thin out. The traces of nervous, muscular, internally organized animals grow more prominent. The first large life had its moment, and then, in many forms, disappeared. What comes next is the part of the story that made the Cambrian famous. Not just big bodies on the seafloor, but bodies with teeth, claws, hard parts, and complex guts. Ecosystems where being large and soft is suddenly a liability. and where evolution begins an arms race that will ripple through the rest of animal history. If you just flip through a geology textbook, the transition looks brutal. One page: strange quilted mats, fronds, and faint little burrows in late Ediacaran mudstones. Next page: shells, spines, complex eyes, predators, burrow systems like subway maps, and almost every major animal body plan you can think of crammed into a handful of rock layers labeled early Cambrian. It's no wonder people look at that and say, wait, did all the animals just appear out of nowhere? That apparent jump is what we call the Cambrian Explosion. But explosion is a bit misleading if you imagine a single instant, a geological Big Bang where life goes from zero to sharks in the span of a storm. In reality, the Cambrian Explosion is more like a fireworks show that runs for tens of millions of years. On human timescales, that's incomprehensibly long. On Earth history timescales, it's a blink. Most geologists bracket the classic Cambrian diversification between about 541 and 520 million years ago, with important build-up and aftershocks stretching from roughly 555 down to maybe 505 million years ago, depending on which signals you care about. So, not instant, but also not the slow, almost glacial pace of earlier evolution, where major changes in ecosystems could take hundreds of millions of years. What makes the Cambrian stand out is the rate and the kind of change. Within a relatively tight slice of time, we see the first clear fossil evidence of complex, internally segmented bodies, well-developed heads and tails, eyes and other sensory structures, specialized front ends with jaws, claws, or grasping appendages, hard parts, mineralized shells, spines, plates, and internal supports, deep branching burrow systems showing active, three-dimensional use of the seafloor. And crucially, we see representatives of most of the big animal branches that still dominate today: arthropods, mollusks, brachiopods, echinoderms, chordates, sponges, conidarians, and a whole riot of weirdos that look like they're hovering near the bases of those groups, or often side experiments of their own. To a first approximation, the Cambrian Explosion is when the tree of animal life suddenly becomes visible. But that's not the same as saying it's when the tree of animal life suddenly began. This is where the rock record plays a nasty trick on intuition. Sediments don't pile up as a perfect continuous tape. They're more like a stack of surviving newspaper clippings from a city that's mostly burned down, scattered, gappy, biased toward some districts over others. If you only found two clippings, one reporting an empty field and a later one showing a built-out metropolis, you might conclude the city popped up overnight. You'd be wrong. You just missed all the intermediate issues. Something similar happens in the late Ediacaran and early Cambrian. Before the Cambrian, most organisms are entirely soft-bodied and lightly built. They live in ways that don't guarantee preservation. They die, decay, and vanish into the mud without leaving sturdy parts behind. Even when they do fossilize, you need just the right conditions to capture them: fine sediment, low oxygen, rapid burial, minimal disturbance. Those just right conditions are rare. Then, right at the Ediacaran-Cambrian transition, several things change at once. Animals start experimenting with hard parts: little tubes, plates, spicules, shells. Burrowers churn the sediment. Ecosystems get more active and three-dimensional. And in a few lucky places, you get sedimentary cameras that freeze those communities in fine detail. This combination makes it look as if complex animals were simply switched on. In reality, the camera just finally starts recording. Think about two different kinds of evidence paleontologists use: body fossils, actual remains, shells, bones, impressions of soft tissues; trace fossils, the footprints and graffiti of life, burrows, tracks, feeding trails. The trace fossil record of animals starts shifting before the classic Cambrian body fossil explosion. Remember those late Ediacaran wiggles and surface grazes in the microbial mats? By the very latest Ediacaran and earliest Cambrian, those traces become more complex. Looping, branching tunnels, deeper tier burrows, vertical shafts going down and back up. That tells us that animals capable of fairly sophisticated movement, sensing, and sediment engineering were already there, even if their bodies weren't being nicely preserved. In other words, behavior gets more complex before bones and shells become common. So why do the Cambrian rocks look so abrupt? Three big reasons. First, rock resolution. For deep time, a layer can represent thousands to hundreds of thousands of years. When you zoom out to that scale, a gradual diversification over 20 or 30 million years compresses into what looks like a single thick line on a stratigraphic chart. Second, sampling. Most of the planet's sedimentary rocks from this age are buried, eroded, or never formed in the first place. The handful of places we do have, Australia, China, Canada, a few others, carry an outsized share of what we know. New finds can radically change the story because we're reconstructing a global revolution from just a few local windows. Third, preservational windows, special kinds of deposits that do an unusually good job of capturing soft-bodied life. The most famous of these are Cambrian Lagerstätten, literally storage places in German, fossil hoards. Two in particular anchor our picture of the early animal world. Xinjiang, in southwest China, dates to about 518 million years ago. Its mudstones preserve an entire seafloor community in extraordinary detail. Worms with bristles, early arthropods with feathery limbs, possible early chordates with notochords and muscle blocks, jelly-like creatures, sponges, things with spines and scales and bizarre feeding appendages. The Burgess Shale in the Canadian Rockies is a bit younger, about 508 million years old, but shows a similarly stunning snapshot. Anomalocaris with its spiny graspers and ringed mouth, Opabinia with 5 eyes and a vacuum cleaner snout, Morella with delicate spines, Hallucigenia walking on spikes with a fringe of soft tentacles along its back. These aren't just isolated freaks. They come with a background cast: algae, mud, microbial films, simple worms, shelly creatures, and predators all mixed together. Xinjiang and the Burgess Shale let us see the invisible majority of soft-bodied animals that almost never show up in the more ordinary fossil record. Without them, the Cambrian would look like a sudden appearance of shells and teeth. With them, we realized there was already a deep bench of soft-bodied diversity. Those sites are snapshots, not the whole movie. They show us that by the time you get into the Middle Cambrian, animal ecosystems are already highly structured, with multiple trophic levels, complex food webs, and a bewildering array of body plans. So when we talk about the Cambrian Explosion, what we really mean is, this is the interval where many major animal lineages show up with clear, easily recognizable fossils for the first time, not necessarily the interval when those lineages first evolved. Take arthropods, the group that includes modern insects, spiders, crustaceans. In the fossil record, their big obvious forms, trilobites, large swimming predators, armored crawlers, appear early in the Cambrian and diversify fast. If all you had were mineralized exoskeletons, you might think arthropods popped into being right there. But if you look at molecular clock studies, genetic analyses that estimate when lineages split based on DNA differences, and combine them with the trace fossils, you get a different picture. Many researchers infer that the deepest roots of the arthropod lineage, and of other major animal groups, go back into the late Ediacaran or even somewhat earlier. Those early members might have been small, soft and simple, easy to miss, hard to preserve. The first fossil appearance is not the birthday of the group. It's just the first surviving photograph. This distinction matters when people try to use the Cambrian Explosion as a mystery to be explained, or as something that supposedly breaks evolution. Once you remember that, the timeline is tens of millions of years, not a single moment. The fossil record is patchy and biased. Behavior and soft-bodied diversity precede obvious hard parts. Molecular data suggests lineages diverge before their first body fossils show up. The suddenness becomes less about life violating evolutionary rules and more about geology finally giving us usable evidence. That doesn't mean there's nothing to explain. There clearly is. Why did so many lineages start experimenting with new body plans, new sizes, and new ecologies in this window? Why do skeletons and shells proliferate when they do? Why does predation intensify? Why do burrows suddenly deepen and ramify? Why does the seafloor shift from a quiet mat world to a churned-up, bioturbated landscape? Scientists have proposed a whole suite of contributing factors: rising oxygen levels, changes in ocean chemistry, the evolution of developmental toolkits that made it easier to generate new body structures, ecological feedbacks from predation and competition, even the breakup of supercontinents changing nutrient flows. In the next sections, we'll pull some of those threads apart. We'll look at how predators and prey may have locked each other into an evolutionary arms race, how genetic and developmental innovations made wild new forms possible, how the appearance of hard parts both reflects and distorts what we see in the rocks, and how different lines of evidence, fossils, chemistry, DNA, sometimes agree and sometimes seem to argue about when the explosion really began. For now, keep one idea in mind. The Cambrian explosion wasn't magic, and it wasn't nothing. It was a real, accelerated diversification of animal life. But what exploded in the rocks had been building quietly and invisibly for a long time. If you really want to understand why the Cambrian feels different, zoom in on one relationship. Who's eating whom? For most of the history we've talked about so far, complex active predation, one animal hunting down and consuming another macroscopic animal, seems to have been rare or limited. Ediacaran seafloors look like gentle worlds. Filter feeders, mat grazers, osmosis specialists, maybe some slow, soft-bodied browsers nudging their way through microbial carpets. Then, across the Ediacaran-Cambrian transition, the tone changes. Suddenly, the rocks start whispering a new story, a violent one. Predators don't just add another character to the cast, they rewrite the entire script. Once you have animals that can chase, grab, crush, and tear other animals, everything about an ecosystem shifts. Where organisms live, how they move, what they build their bodies out of, how fast they grow, how many offspring they produce, how complex their nervous systems become. Predation doesn't just kill prey, it creates pressure. Constant directional creative pressure. Evolution listens. We can actually see that shift in the fossils. Early Cambrian rocks and a few very latest Ediacaran beds start showing signs that somebody out there was not just grazing on mats, but actively attacking other animals. You get small circular drill holes in shells and tubes, neatly bored, often right where they'd hit soft tissue inside. That's not random damage. That's a predator with a plan, using some kind of rasping or chemical toolkit to perforate a hard surface and suck out the juicy bits. You get shells and exoskeletons with healed fractures, cracks and chips that partially regrew. That tells us the animal survived an attack, lived long enough to repair the wound, and carried that scar into the fossil record. You get certain kinds of arthropod appendages, jointed, spine-covered, shaped like grappling hooks or bear traps. These aren't delicate paddles for gentle swimming. They're built to seize, hold, and manipulate struggling prey. In animals like Anomalocaris, you see paired frontal appendages covered in inward-pointing spines, perfectly designed to grab and funnel prey into a circular, tooth-lined mouth. You see body plans that only make sense if something is trying to avoid being eaten. Trilobites with thickened cephalon shields and rolled-up defensive postures. Tube-dwelling worms encased in mineralized armor. Tiny shelly creatures, small shelly fossils, forming protective houses out of calcite or phosphate. In other words, the Cambrian explosion is not just an explosion of forms, it's an explosion of ecological roles, especially at the sharp end of the food web. Once predators arrive in force, prey don't have the option of ignoring them. They either escape, hide, defend, or die. One of the most dramatic responses is biomineralization, building hard parts out of minerals pulled from seawater, calcium carbonate, calcium phosphate, silica. That shift is doing at least 3 jobs at once. First, defense. A soft, squishy body is easy pickings for any animal with teeth, claws, or a strong enough suction. Wrap that same body in a rigid shell, stud it with spines, or plate it with armor, and suddenly many attacks bounce off, or at least become much more costly for the predator. Second, structure. Hard skeletons and exoskeletons don't just shield, they support. They let you build bigger bodies, stronger muscles, more elaborate appendages, jaws, teeth, claws, internal rods to anchor swimming muscles, external cases to protect delicate gills or soft tissues. Third, record keeping. Hard parts fossilize far more readily than soft ones. The moment animals start experimenting with shells, spicules, and skeletons, the rock record lights up. Creatures that would once have vanished without a trace now leave piles of mineralized debris. That's part of why the Cambrian turns on so suddenly. But defense doesn't end with armor. Some animals go down, literally. In the late Ediacaran, the seafloor surface is often coated in relatively undisturbed microbial mats and covered by shallow surface traces, wiggles, grazes, little horizontal tubes. The mud below is mostly quiet. By the early Cambrian, that changes dramatically in what paleontologists call the Cambrian substrate revolution. Burrows suddenly start going deep. You get vertical shafts plunging centimeters into the sediment, branching tunnels that twist and loop in 3 dimensions, networks that connect the surface to multiple underground tiers. This isn't just a change in style, it's a change in lifestyle. Why dig down? Hiding from predators is one big reason. If dangers patrol the surface, swimming arthropods, crawling hunters, grasping tentacle things, it pays to live underneath the killing zone. Burrows also let you exploit new food sources, organic particles buried in the mud, bacteria growing inside the sediment, oxygen gradients in pore waters. But as soon as you have animals bulldozing, mixing, and aerating the upper layers of the seafloor, you transform the environment for everyone else. Those once stable microbial mats get ripped up and eaten. Fine layering in sediments is destroyed. Oxygenated water is pumped down into deeper layers, changing redox conditions and nutrient cycles. In modern oceans, bioturbation, the churning of sediment by animals, is a big driver of seafloor chemistry. The Cambrian marks the first time in Earth history that animal behavior systematically engineers the substrate on a global scale. Predators above the sediment force prey into it. Prey in the sediment rework the seafloor. The new seafloor chemistry feeds back into what kinds of skeletons and shells are chemically easy or hard to build. It's all connected. And once predation, armor, and sediment engineering get going together, you get feedback loops. Imagine an early Cambrian shoreline. At first, you have simple grazers and filter feeders. A slightly more active carnivore evolves, maybe an arthropod that can nip and shred soft tissues. Now, any mutation that makes prey a bit harder to eat, slightly thicker body wall, the first thin mineral coating, the habit of burrowing when disturbed, gets favored. As more individuals evolve better defenses, the old weak predators start to struggle. Some lineages respond by going extinct. Others push back. They evolve stronger mouthparts capable of crunching thin shells. They develop better eyes to spot buried prey. They lengthen and articulate limbs to probe into burrows or pluck armored creatures off the bottom. Those improved predators raise the bar again. Now, only prey with even better armor, deeper burrows, faster escape responses, or more sophisticated camouflage really thrive. Repeat this dance, generation after generation, over millions of years, across many lineages. What you get is an evolutionary arms race, not between 2 species, but across entire communities. Predators select for better defenses. Better defenses select for more powerful, precise, and intelligent predators. At the same time, competition within each side matters. Prey species compete for food and safe space. Predators compete for prey. Any tweak that lets you use a slightly different resource, dig a slightly different depth, graze at a slightly different time of day, specialize on a different shell thickness, can carve out a new niche. That opens up ecological room for more species to coexist. So predation doesn't just kill individuals, it generates diversity. Importantly, most researchers don't think predation was the single trigger of the Cambrian explosion. It's more like an amplifier. Oxygen levels rising enough to support high-energy lifestyles make active predators physically possible. Changes in seawater chemistry make biomineralization easier. Genetic and developmental toolkits, which we'll talk about next, make it feasible for animals to build new structures, eyes, limbs, shells, without reinventing biology from scratch. Against that backdrop, as soon as a few lineages stumble into effective predatory lifestyles, the game intensifies. You get the origin and elaboration of eyes capable of forming images, not just sensing light. More complex nervous systems for tracking moving targets and coordinating escapes. Hard parts both for offense, teeth, claws, and defense. Shells, spines, plates. Burrowing, tube building, reef forming, and other environmental engineering that changes the rules for everyone. These are not isolated quirks. They're cascading responses in a network. The Cambrian explosion, seen through this lens, isn't just about new animals showing up. It's about animals discovering each other as enemies, resources, and ecosystem engineers, and then dragging each other, step by reluctant step, into new evolutionary space. You can think of pre-Cambrian life as mostly 2-dimensional. Mats and surfaces, slow grazers, gentle filters. By the mid-Cambrian, life has gone fully 3-dimensional. Water column, seafloor, and subsurface are all packed with interacting, evolving creatures. Predators slicing through the water, burrowers tunneling below, armor-clad grazers trundling across the top, And running under all of it is a quieter revolution. Changes inside cells and embryos that make such rapid experimentation with body plans possible in the first place. To understand why the arms race could escalate so fast, why eyes, limbs, shells, and spines could proliferate across so many different groups, you have to look under the hood of animal development. That's where we go next. Imagine you could zoom in on a Cambrian animal, not from the outside, but from the very beginning, from the first few hours after an egg is fertilized. To your naked eye, it's just a tiny blob of cells, dividing, folding, rearranging. But inside that blob is a choreography so intricate that if you saw it play out on a screen, you'd swear it was pre-programmed by some master engineer. Head here, tail there, mouth in this region, limbs sprouting from this zone, not that one. Nervous system bundling along the back, gut tunneling through the middle. The question is, how? What changed between the simple, mostly flat, soft-bodied Ediacaran organisms and the riot of articulated, segmented, eye-studded, armor-clad creatures of the Cambrian? Part of the answer is ecological, predators, oxygen, chemistry, all the stuff we've just talked about. But there's another part that lives inside embryos. It's not just about having the right genes, it's about how you use them. Biologists sometimes call this field Evo-Devo, evolutionary developmental biology. At its core is a simple but powerful idea. If you change when, where, and how strongly genes turn on during development, you can reshape the body without inventing completely new genes every time. The raw materials are already there. Evolution just rewires the control panel. You can see this remarkably clearly in a set of genes that became famous in the late 20th century, the Hox genes. In fruit flies, Hox genes are arranged on the chromosome in a very particular order. When the embryo develops, those genes switch on in overlapping bands along the head-to-tail axis. Gene at one end of the cluster, active near the head. Gene at the other end, active near the tail. The ones in between, various zones in between. Each band doesn't build a specific organ by itself. It sets up identity. This region will be head-like. This region will be thorax-like. This region will be abdomen-like. Other genes then fill in the details. What kind of limb? What kind of segment? What specific tissues? When researchers looked at very different animals, mice, chickens, even humans, they found something astonishing. The same basic Hox toolkit was there. A cluster of genes turned on in a staggered pattern along the body axis, helping define head, neck, trunk, and tail regions. In other words, wildly different creatures share a deeply conserved body map system. The implication for the Cambrian is subtle but profound. Before the explosion of animal forms, the genetic machinery needed to lay out a complex body, front to back, top to bottom, was already coming together. You don't need a brand new Hox cluster every time you evolve a new body plan. You need new ways of deploying the cluster you have. Shift the boundary where one Hox gene turns off and another turns on, and you can lengthen one region at the expense of another. Tweak how certain downstream genes respond to a particular Hox combination, and you can turn a simple lateral flap into a jointed limb or a gill-bearing branch. Gene-wise, that's a modest edit. Morphology-wise, it can be a revolution, and Hox genes are just the headline act. Animals also rely heavily on regulatory DNA, stretches of sequence that don't code for proteins at all but act like switches and dimmers. A typical tool gene, say, a gene involved in making a limb, can have dozens of these regulatory modules scattered around it. One module might say, turn this gene on in the developing leg, but only in the upper portion. Another might say, turn this gene on in the antenna, but only in this precise pattern. Another, switch this gene on briefly in the embryonic spine, then shut it off forever. Here's the clever part. You can evolve changes in one regulatory module without wrecking all the others. That means you can tinker locally, alter a limb, lengthen a fin, add spines to a specific segment, without destroying functions in other tissues. This is modularity. Bodies are built in semi-independent pieces, segments, appendages, organs, coordinated by a shared genetic language, but each controlled by its own local regulatory scripts. From an evolutionary standpoint, it's like having a machine assembled from replaceable parts, instead of a solid block you have to recarve every time you want to tweak. Now add one more ingredient: gene duplication. Every so often, the gene gets copied by mistake. Instead of one copy of a developmental regulator, you have two. At first, both do the same job. But over time, one copy might stay conservative, preserving the original function, while the other is free to mutate and specialize. That's how you get families of related genes that all share a core function, patterning the body, shaping limbs, regulating cell death, but attuned to different contexts. In vertebrates, for instance, the Hox cluster itself duplicated multiple times. We carry several Hox clusters, each with related but distinct roles. The same principle applies to many other developmental regulators, transcription factors, signaling molecules, receptors. By the time you hit the late Ediacaran and early Cambrian, the ancestors of modern animal groups seem to have accumulated a pretty rich set of these duplicated, diversified control genes. So picture the situation going into the Cambrian arms race. A head-to-tail body map system, Hox and friends, that can label regions. A suite of signaling pathways like WNT, BMP, Hedgehog, Notch, that can carve up tissues into patterns and boundaries. Regulatory DNA modules that let each body region deploy these signals in different combinations. Duplicated genes providing raw material for new functions, eyes, limbs, shells, specialized cell types. Under calm ecological conditions, a lot of this potential could remain mostly latent. You might get slow, incremental changes in body shape, most of which leave little fossil trace, especially if the creatures are soft-bodied. But once you inject strong, sustained selection, predators, moving sediments, new microhabitats, fluctuating oxygen, this latent flexibility becomes explosive. Every time a small regulatory tweak produces an embryo with slightly thicker cuticle, or a slightly deeper burrowing habit, or a marginally better positioned eye, that individual has a better shot in a predator-rich world. Because the underlying toolkit is modular, those beneficial tweaks don't necessarily wreck everything else. The animal is still viable. It just has a new variation on an existing theme. This makes morphological experimentation cheap. Not free, most mutations are still neutral or harmful, but cheaper than if every change required inventing a brand new protein from scratch. Over millions of years and across many lineages, cheap experimentation means you see a lot of different body layouts explored, different segment counts, eye placements, limb arrangements, armor schemes. Most of those experiments vanish. Some leave fossils we can admire. A few become the founding plans of major modern groups. Still, there's an important caveat. The genetic toolkit doesn't cause the Cambrian explosion by itself. Many of the key toolkit components likely evolved well before the Cambrian, possibly hundreds of millions of years earlier, as early animals branched off from their single-celled relatives. Sponges and simple cnidarians, like jellyfish and sea anemones, already show traces of some of these regulatory systems, though often in simpler forms. The toolkit is more like a set of possibilities waiting for the right ecological and environmental conditions to make them pay off. You can have the capacity to build complex eyes long before it's actually favored by selection. If there's no bright, predator-filled water column to look into, elaborate vision might never evolve. You can have the machinery to articulate limbs long before you see truly jointed walking legs. If the seafloor is covered in stable microbial mats with no deep burrows, there may be little advantage in powerful digging or grasping. The Cambrian explosion is what happens when pre-existing developmental flexibility collides with a world that suddenly rewards and punishes body design. In that sense, the 3 billion years of nothing line is doubly misleading. First, because there was a lot happening in those billions of years. The origin of life, photosynthesis, oxygenation, eukaryotes, early multicellularity. Second, because much of the genetic and developmental groundwork for animals was being laid quietly, in creatures that either didn't fossilize well or left only ambiguous traces. By the time shells, spines, and articulated skeletons burst into view, the cellular machinery that made them possible had been evolving in the background for a very long time. That helps explain why the fossil record can look like a sudden jump. It's recording the moment when developmental possibilities finally meet ecological demand and mineral-friendly seawater. Not the birth of complexity from nowhere, but the unveiling of complexity that had been slowly assembled and then rapidly deployed. So when you look at a Cambrian trilobite with its many segments, jointed legs, faceted eyes, and rigid exoskeleton, you're seeing the visible surface of a much deeper story. Hox stripes quietly labeling body regions, signaling gradients carving up the embryo, regulatory modules turning shared genes on and off in novel patterns, duplicated developmental switches repurposed for new structures. Evolution didn't have to invent trilobite-ness from a blank slate. It had a flexible, modular construction kit, and a world that suddenly demanded armor, movement, and perception at a higher level. In the next part of the story, we'll see how one outcome of that combination, the invention of skeletons in particular, did something else remarkable. It didn't just change how animals lived, it changed how we can see them at all. Picture the seafloor just before the Cambrian really kicks off. Most of the animals there are still soft. Wormy bodies, gelatinous bags, maybe a bit of tougher outer skin, but nothing you'd mistake for a shell. If you could time travel and snorkel through that world, you'd see a lot of life. But if you came back and tried to find that same life in the rocks, almost nothing. That's the brutal reality of the fossil record. Nature does not hand out participation trophies. It plays favorites, and one trait in particular stacks the deck: hard parts, teeth, shells, spines, plates, tubes. Anything made from minerals like calcium carbonate, calcium phosphate, or silica. Soft flesh rots, squishes, and disappears unless it's buried in just the right way, in just the right mud, under just the right chemistry. But a mineralized shell that can sit on the seafloor for years, get buried, and still have a fighting chance of turning into rock. So when animals start building skeletons, two things happen at once. Ecologically, they change the game. Predators can bite harder. Prey can armor up. Burrowers can dig more aggressively. Geologically, they suddenly become visible. The camera turns on, and the very first flicker of that camera isn't big trilobites or dramatic armor plates. It's something far more modest and far easier to miss. Tiny fragments, millimeter-scale bits of mineralized stuff. This is the world of the small shelly fossils. If you sift through late Ediacaran and very early Cambrian rocks with enough patience and a good microscope, you start finding them. Little tubes, cones, spicules, plates, and weird, gear-looking pieces. Many are made of calcium phosphate or carbonate. Some are bits of early skeletons. Some might be protective capsules for larvae. Some are parts of organisms we still can't identify. They're so small that when paleontologists first started noticing them in the mid-20th century, they didn't look like much, just crumbs. But those crumbs carry a huge message. They tell us that biomineralization, the habit of grabbing dissolved minerals from seawater and turning them into hard body parts, was already underway before the Cambrian Explosion hits its visual peak. We're not going from a world of pure jelly to fully armored trilobites overnight. We're seeing a ramping up, scattered experiments in hard parts, gradually spreading and diversifying. Some of these tiny fossils likely belong to stem groups of brachiopods, mollusks, sponges, and other early animals. Others may be the detached scales and plates of creatures whose full bodies we almost never see. And that almost never is important. Because even with skeletons, fossilization is still rare. For most of Earth's history, the default fate of a dead organism is to vanish without a trace. To get a fossil, you need a coincidence: rapid burial, low oxygen, the right microbes, the right chemistry. To get soft tissues preserved, the guts, limbs, eyes, and delicate bits, you need an even more extreme coincidence. Every so often, the planet gives us that coincidence in spectacular fashion. Paleontologists call these sites Lagerstätten, German for storage places, and they're the reason we can see Cambrian life in such detail. Think of the Burgess Shale in Canada, or the Zhenjiang Biota in China. These are places where muds, chemistry, and burial conditions lined up so perfectly that even unmineralized tissues got preserved as thin carbon films or fine mineral replacements. Without these exceptional deposits, our picture of early animals would be almost entirely shells and spines, interesting, but incomplete. With them, we suddenly see the soft-bodied underworld. Worms with elaborate feeding appendages, slug-like creatures with rows of spines, arthropods with complex eyes and feathery limbs. Lagerstätten let us peek behind the armor and see what the Cambrian toolkit was actually doing with bodies. But there's a catch. Because these sites are so visually dramatic, they can give us a skewed impression. It's easy to look at a Burgess Shale slab and think, wow, all this complexity appears right here, right now. In reality, we're looking at a snapshot where conditions just happen to preserve everyone. Not necessarily the moment when everyone evolved. This is where we have to talk about an unglamorous but crucial idea: fossil bias. The rocks are not a neutral record, they're a filter. First, there's the basic filter of who gets fossilized: hard parts over soft parts, buried over unburied, quiet seafloors over wave-swept beaches. Then there's the filter of what rocks we still have. Most of Earth's crust has been recycled, metamorphosed, or eroded away since the Proterozoic. The older you go, the fewer undisturbed sedimentary rocks are available to even look at. And on top of that, there's the filter of where humans have actually searched. Some regions are intensively studied, others are barely touched. Some ages have lots of accessible outcrops, others are tucked under oceans, jungles, or cities. All those filters pile up and create illusions. One of the clearest of those illusions has a name, the sinulipse effect. Here's the idea. Imagine a species that actually originated 550 million years ago and went extinct at 540 million years ago. In an ideal world, you'd find fossils exactly spanning that range. In the real world, you might only find fossils at 547 and 542 million years ago, because that's where rocks of the right type happen to be preserved and exposed. Or maybe your oldest fossil is at 544, just because earlier sediments weren't preserved, or you haven't found them yet. If you look only at the data you have, you'd conclude the species appeared later than it really did, and disappeared earlier than it really did. The first and last appearance in the fossil record are almost always late arrivals and early exits compared to the actual history of the lineage. Scale that up from one species to dozens or hundreds, and you can see the problem. A genuine, gradual diversification can look artificially abrupt if the earlier parts are poorly preserved, undersampled, or locked in difficult rocks. Early members of major Cambrian groups, arthropods, mollusks, echinoderms, may have been small, rare, or soft-bodied. Their fossil footprints are faint. They only become obvious once they evolve hard parts, spread widely, and start dying in large enough numbers that some fraction get preserved. So when we see the fossil record switch on around the base of the Cambrian with a sudden bloom of hard parts and diverse forms, part of that bloom is real biological change. But part of it is the sinulipse effect and its cousins. Fossils can't show us what wasn't preserved, and early subtle phases of lineages are easier to miss. Rock availability plays in too. Late Ediacaran and early Cambrian marine sediments happen to be relatively abundant and well preserved in many parts of the world. Go much earlier, and the suitable rocks thin out, get more metamorphosed, or are buried under later deposits. If you had a time machine and could walk along the seafloor at, say, 560 million years ago, you might see a modest but real level of diversity. Small bilaterian worms, simple grazing forms, maybe proto-mollusks scraping microbial mats. But because there are fewer rocks from that time, and fewer animals with hard parts, their apparent diversity in the fossil record is much lower. Put all this together and you get an uncomfortable but important conclusion. The fossil record is not a straight photograph of evolution. It's more like a series of patchy security camera stills, taken at irregular intervals with motion blur and whole hours missing. Does that mean the Cambrian explosion is just an illusion? Most researchers don't think so. Even after you correct for known biases, sampling intensity, rock volume, preservational quirks, you still see a marked increase in the number of distinct animal groups and skeletal innovations through the early Cambrian. Small shelly fossils ramp up in abundance and complexity over time, not just in one place, but across multiple continents. Skeleton types diversify. The range of burrows carved into sediments expands dramatically, suggesting new modes of life below the seafloor. Trace fossils of movement, trackways, feeding trails, become deeper, more complex, and more widespread. And crucially, those exceptional Lagerstätten from China, Canada, Greenland, and elsewhere all tell a broadly consistent story. By about 520 to 515 million years ago, we're looking at ecosystems with representatives of most major animal body plans already present. Early arthropods, preapulid worms, caudate-like swimmers, stem echinoderms, stem mollusks, and more. Not random one-offs, but whole food webs with multiple tiers of predators and prey. Biases can sharpen the apparent edge of the explosion, making it look more like a cliff than a steep slope. They can hide the roots and the earliest branches, but there's still a real, accelerated branching there. A period where the combination of ecological pressure, developmental flexibility, and seawater chemistry produced an uptick in both innovation and fossil visibility. From our point of view, skeletons are the turning point. They didn't just protect Cambrian animals from each other, they protected their memories from geological oblivion. They let lineages write in ink instead of pencil, and once that writing appears, first as scattered small shelly bits, then as full shells and articulated exoskeletons, we finally get to trade speculation for evidence. We can start timing when different animal groups show up, comparing those times to genetic estimates of divergence, and asking a deeper question. How much of the Cambrian explosion is about evolution speeding up, and how much is about our window on evolution suddenly clearing? That's where we go next, when we put molecular clocks and rocks side by side and see how well their stories line up. So on one side, we've got the rocks. They're messy, biased, incomplete, but they're physical. You can hold them in your hand. You can date the volcanic ash layers above and below them. You can point to a trilobite and say, this existed by this time, no later. On the other side, we've got something much more abstract, the molecular clock. Instead of reading time from layers of mud and ash, molecular clocks try to read it from DNA. Here's the basic idea in plain language. Every time a cell copies its DNA, it makes tiny mistakes, mutations. Most don't do much. A lot of them accumulate in places that don't strongly affect the organism's survival. Over many generations, these changes pile up. If two lineages split from a common ancestor and go their separate ways, their DNA starts to drift apart. The longer they've been separated, the more differences build up between their genomes. If you know roughly how fast those differences accumulate, how many DNA changes per million years, you can use the amount of difference between two species today as a kind of genetic stopwatch. That's the clock. Count the differences, divide by the rate, get an estimate of when their lineages split. In principle, this is powerful. Even if the rocks are silent, the genomes of living animals might still carry a blurred record of when their ancestors diverged. But there's a catch. Actually, several. Molecular clocks are not like the quartz watch on your wrist. They don't tick at a perfectly steady rate. Different genes mutate at different speeds. Different groups of organisms can speed up or slow down their rates over time. Life histories, body size, metabolism, all of that can tweak how quickly mutations accumulate. So when scientists build a molecular clock, they have to make choices. Which genes do you use? How do you model the fact that rates can change between lineages? Do you assume one global rate, or do you let each branch of the tree have its own tempo? And crucially, how do you calibrate it? Because the molecular clock doesn't know about years on its own. It only knows number of mutations. To turn that into millions of years, you need at least a few points where you already have a good idea of the real age. Usually those calibration points come from fossils. For example, if you have a nice, well-dated fossil that clearly belongs to an early member of the vertebrate group, you can say, okay, by this time, the vertebrate lineage must have already split from its closest relatives. You plug that as a minimum age into your molecular analysis. Do that for many branches, and the clock has something to anchor to. Change the calibrations, though. Use a different fossil, interpret its placement differently, or revise its age, and the whole clock can shift. Rates, genes, calibrations, and the exact mathematical model all interact. That's why, if you look across the scientific literature, you'll see a frustrating range of molecular clock dates for the origin of animal groups. Some analyses put the main splits between major animal lineages, things like the divergence of sponges from other animals, or the separation between cnidarians, jellyfish and their kin, and bilaterians, most of the rest of us, way back in the deep Neoproterozoic, 800 million years ago, 900 million, in some early studies, even flirting with 1 billion years. Others, using different calibrations and models, pull those dates forward, closer to the Ediacaran and early Cambrian, say, 650 to 550 million years ago for the key branches. What happens when you lay these clock estimates over the rock record? You get a tension that has driven 2 decades of debate. Because the fossils tell a story where obvious complex animals with clear bilateran body plans show up mostly in the latest Ediacaran and then really take off in the early Cambrian, between about 575 and 520 million years ago. But many molecular clocks say the lineages that would give rise to those animals had already split apart tens to hundreds of millions of years earlier. In other words, the family trees were in place long before their major branches leave bones and shells in the rocks. Take arthropods, the group that includes insects, spiders, crustaceans, and their Cambrian cousins. The fossil record gives us clear arthropod-like forms by the early Cambrian, and trace fossils that might hint at arthropod ancestors a bit earlier. Some molecular clocks, though, push the arthropod lineage back into the mid-Neoproterozoic. Same for mollusks, annelid worms, and other big animal groups. Clocks often say their last common ancestors were already around well before the Cambrian Explosion's visible fireworks. So what do we do with that mismatch? There are a few broad options. One is, the clocks are off. Maybe mutation rates weren't as constant as some models assume. Maybe some lineages went through periods of faster or slower change. Maybe some calibrations used fossils that were misinterpreted or misdated. We know this happens. When key fossils get reevaluated, or new older fossils show up, molecular clock estimates often get recalibrated and pulled closer to the fossil record. Another option is, the rocks are lying by omission. Maybe those early members of the major animal lineages really were there, hundreds of millions of years before the Cambrian, but we just haven't seen them because they left almost no fossils. This is where ideas like cryptic early animals come in. Imagine an early bilaterian worm living 650 million years ago. It's a few millimeters long. It lives in the sediment or glides over microbial mats. It's soft-bodied, no shell, no spines. Its population sizes are small. Its geographic range patchy. It doesn't build reefs or carve dramatic burrows. Its body is just a bit more complex than the simpler worm-like creatures around it. From the point of view of the rocks, that animal is almost invisible. Its soft tissues decay fast. The sediments of that time are fewer, more altered, more likely to have been squashed and cooked by later tectonics. Even if some individuals did get preserved, they'd be tiny and nondescript, easy to miss in a world where we've only sampled a fraction of ancient rocks. Scale that up. Multiple lineages of small, soft, low-impact animals diverging and quietly exploring body plans for tens of millions of years, all while leaving almost no recognizable trace. Then, as ocean chemistry changes, oxygen levels rise in more basins, and ecological arms races heat up. Some of those lineages start to evolve larger size, tougher tissues, early skeletons, and more vigorous burrowing and grazing. They start to reshape seafloor ecosystems in ways that are much more likely to leave traces. Deeper burrows, disrupted microbial mats, biomineralized bits that resist decay. Suddenly, the cameras that have always been very bad and very patchy at recording soft, rare, small animals become much better at recording these new, more robust, more numerous, more ecologically disruptive forms. From this perspective, molecular clocks and rocks aren't necessarily contradicting each other. They're just looking at different thresholds. The clocks might be telling us when lineages genetically split. The fossils might be telling us when those lineages became large, abundant, and sturdy enough to reliably enter the geological record. Another reconciliation idea is that the earliest animals in those deep clock dates may not have looked like animals in the way we picture them. They might have been simple, flat, ciliated layers, more like beefed-up single-cell colonies or thin tissues, without organs, guts, or muscles in the modern sense. Early experiments in multicellularity that only later coalesced into the complex three-dimensional bodies we recognize as Cambrian animals. If that's true, then saying animals existed at 800 million years ago could be technically correct in a genetic sense, while still leaving room for the familiar active bilaterian animals to only appear much later in fossils. There's also a more mundane truth. Both records, molecular and fossil, have error bars, big ones. Clock estimates often come with uncertainties of tens of millions of years on either side. Fossil first appearances are minimum ages, not precise moments of origin. When you factor those uncertainties in, the apparent clashes sometimes soften into overlaps. For example, a molecular clock might say, this divergence probably happened around 700 million years ago, plus or minus 60 million. The oldest plausible fossil hints of that group might be around 560 million years old, with their own dating uncertainties. That's still a gap, but not necessarily a fatal contradiction. It's a target zone for where the missing record might lie. So where does the debate stand now? Many recent clock studies that use more realistic models, allowing mutation rates to vary, using multiple calibrations tied to better understood fossils, and explicitly accounting for uncertainty, tend to cluster the core diversification of crown group animals somewhere in the Neoproterozoic, probably between about 650 and 550 million years ago. That's before the classic Cambrian explosion, but not absurdly far before the earliest Ediacaran fossils and trace fossils that hint at bilateral activity. In that picture, there's time for a cryptic history of small, soft, increasingly complex animals that slowly diversify in the late Neoproterozoic, followed by a visible ecological and skeletal explosion across the Ediacaran Cambrian boundary. Not 3 billion years of nothing, then instant trilobites, but also not all animal history compressed into a 5 million year window. What could help narrow this down? More and better fossils, especially from older Neoproterozoic rocks, 570, 580, 600 million years old and beyond. Each new Lagosteta from that window has the potential to either confirm or constrain the molecular estimates. Better geochronology helps too, more precise radiometric dates on key fossil-bearing layers, so we know exactly how old those rocks really are. On the molecular side, larger genomic datasets, especially from weird modern animals, sponges, comb jellies, obscure worm groups, help refine the tree of life and the rates along its branches. New methods that model rate changes more realistically and test different calibration choices can show which timelines are robust and which collapse when assumptions change. And there's a deeper conceptual point. We don't have to pick a winner between cocks and rocks. The fossil record shows when and how life became geologically loud, when organisms started leaving traces that reshaped sediments and built skeletons. Molecular clocks try to tell us when the ghost ancestors of those organisms first parted ways in the tree of life, long before they left bones. One record is about bodies, the other is about lineages. Where they agree, we gain confidence. Where they disagree, we get questions. And those questions drive new expeditions, new techniques, and new ideas about how evolution actually unfolds. And that matters, because how we time the Cambrian explosion changes how we think about it. Was it truly a bolt of evolutionary lightning striking a nearly empty sky? Or was it a sudden illumination of constellations that had been quietly forming in the dark for tens of millions of years? Answering that sets the stage for the last part of the story. What happened after the explosion? Who thrived? Who vanished? And what this strange burst of early animal life means for everything that came after, including us. So let's say you could stand on a Cambrian seafloor right after this whole thing goes off. The big splits between animal lineages have already happened. The fossil record has finally woken up. Trilobites are crawling around. Worms are plowing through the mud. Spiny armored swimmers are cruising overhead. What happens next is not a return to calm. The explosion doesn't just flick on and then stop. It keeps echoing. Over the next tens of millions of years, through the rest of the early Cambrian and into the Ordovician, animal life keeps diversifying. Ecosystems keep getting more complex. And evolution keeps throwing out wild new experiments. But there's a pattern. Some of those experiments will end up being the ancestors of everything alive today. Most will vanish. If you were there, you'd see both stories unfolding at once. On one hand, the winners are taking shape, lineages that will eventually give us insects, crabs, clams, fish, starfish, and much later, land vertebrates. On the other hand, the seafloor is full of creatures that look like they crawled in from a completely different universe and then quietly backed out of history. Take the Burgess Shale, that famous Middle Cambrian site in Canada. It's a snapshot of one of these ecosystems maybe 10 or 15 million years after the initial burst. You've got animals we recognize in broad outline, early arthropods with jointed legs, primitive chordates with notochords, sponges, brachiopods anchored to the seafloor. But you've also got hallucination fuel. Opabinia, with 5 eyes on a stalked head and a long grabber snout ending in a claw. Anomalocaris, a meter-long apex predator with rotating pineapple slice mouthparts and 2 spiny arms to shovel prey in. Wiwaxia, a slug-like thing covered in overlapping scales and long spines. Hallucigenia, literally named because early paleontologists thought it had to be a joke. Long tube body, pairs of spines on one side, floppy tentacles on the other, and we weren't even sure which side was up. For a long time, scientists debated whether some of these forms represented totally separate failed phyla, major body plans that had nothing to do with anything alive now. As the fossils and the analyses got better, a more grounded picture started to emerge. Opabinia and Anomalocaris. They're now usually seen as early offshoots of the arthropod line, or close relatives, side branches on the road that leads eventually to insects and crustaceans. Hallucigenia, Turns out it sits on the stem, leading to a modern group called onychophorans, the velvet worms. Still weird, still rare today, but recognizable. In other words, even some of the wildest Cambrian monsters turn out not to be total aliens, but early experimental versions of lineages that mostly did survive, but not all of them. There really are whole groups of Cambrian animals that seem to belong to body plans that just stop. No living descendants we can recognize, no obvious tie-in to later phyla. They appear in a handful of early deposits, hang on for a geological moment, and then get pruned away. That's important. Because when you look at the aftermath of the Cambrian explosion as a whole, what you see is not just an expansion of diversity. You see a winnowing. Early on, evolution is exploring a huge range of possible body plans and lifestyles. Armored slug things, tentacled floaters, weird filter-feeding cones, almost crabs and almost worms and almost vertebrates. Then, as ecosystems mature and competition gets tougher, many of those early experiments disappear. The range of basic body architectures narrows. The crown groups, the lineages that will go on to define modern phyla, start to dominate. By the time you get into the Ordovician, maybe 40 or 50 million years after the core Cambrian heyday, the seafloor looks more familiar. Not in detail. You'd still be confused, but the broad outlines of the food web would make sense. You'd see seafloor engineers, burrowing worms and arthropods constantly churning the sediment. Stationary filter feeders, sponges, bryozoans, early corals, brachiopods, sucking particles from the water. Mobile grazers, snails and other mollusks scraping algae or microbial films. Mid-level predators, trilobites, early cephalopods, arthropods picking off smaller animals. Apex predators, larger cephalopods, big arthropods, later on early jawless vertebrates. Energy flows from microscopic plankton and seafloor microbes up through layers of grazers and filter feeders into specialists that eat them, and finally into top predators. That's a recognizable marine food web. It has niches that echo into today's oceans. Things that act like crabs, things that act like clams, things that act like sharks, even if the exact players are different. So how do we connect those Cambrian groups to the animals around us now? Some ties are straightforward. Arthropods are the poster children. By the mid-Cambrian, you have clearly segmented, joint-legged animals with exoskeletons. Over time, that lineage branches and branches again. Some lines die out, but others give rise to trilobites, huge success in the Paleozoic, then extinct. Chelicerates, line that now includes spiders, scorpions, horseshoe crabs, crustaceans, crabs, lobsters, shrimp, myriapods, centipedes and millipedes, insects, eventually dominating land ecosystems, same with mollusks. Early Cambrian fossils show simple shelly forms and primitive snail-like or clam-like creatures. As time goes on, those lines diversify into gastropods, snails, slugs, bivalves, clams, mussels, cephalopods, squids, octopuses, cuttlefish, and their shelled ancestors like ammonites and nautiloids. Chordates, the group that includes us, also trace back to Cambrian forms. Tiny lancelet-like swimmers such as Bicaia and Hyquichtsthis show early versions of key chordate features: a notochord, a dorsal nerve cord, muscle blocks along the body. From there, jawless fish appear, then eventually jawed fish, then vertebrates invade land. In all these cases, the Cambrian gives you the stem branches that lead into the crown groups we see now. But there are also organisms that are much harder to place. Some are only known from a few squashed impressions. Others mix features we'd usually associate with different phyla. Our phylogenetic tools can sometimes wedge them onto a tree, but the deeper you go, the fuzzier the picture gets. And that fuzziness is telling us something deep about how evolution works. The Cambrian explosion wasn't just a time of invention. It was a time of ruthless selection. Every new adaptation, a better way to burrow, a harder armor plate, sharper vision, more efficient digestion, changed the ecological game for everything else. If one lineage evolved a novel weapon or defense, its competitors suddenly had to keep up or get sidelined. Sometimes they kept up, sometimes they didn't. Extinction becomes just as important as innovation. That's the contingency piece. Imagine rewinding Earth's history to the early Cambrian and letting it run again. A slightly different oxygen curve. A few random storms hitting one basin and not another. a minor meteorite landing in a different ocean. Tiny differences in who survives which local disasters, or who happens to evolve a new trick first, could have shifted which body plans became dominant. Maybe in that alternate history, a group we only know as a weird Burgess shale oddball is the one that takes off, radiates, and produces the future equivalents of fish, insects, and mammals. Our own branch, the chordates, might stay small and obscure, or vanish completely. We tend to talk about the Cambrian Explosion as if its outcome was inevitable. Of course arthropods win, of course vertebrates arise, of course intelligence eventually appears, but the more closely we look, the less that sounds like a law, and the more it looks like one particular path that survived out of many. Evolution is full of these bottlenecks and branching points, long, slow build-ups of potential, genetic toolkits, ecological opportunities, slowly rising oxygen, followed by relatively brief intervals where that potential is subtly expressed in a rush of new forms. The Cambrian is one of the clearest examples, but not the only one. You see echoes of the pattern later, after mass extinctions like the end Permian and end Cretaceous, when surviving lineages rapidly radiate into emptied niches in the origin of land plants and forests, which reshaped climate and soils and opened new evolutionary doors. In the evolution of mammals after the dinosaurs' crash, and again in the rapid spread of hominins with symbolic culture and technology. In each case, a long, often invisible preparation phase is followed by a visible burst of complexity, slow to build, fast to express. That raises a larger question. Is this just how complex systems behave when they're pushed? Do you get long periods where variation and potential accumulate quietly, buffered by constraints, followed by shorter periods where those constraints loosen because of a climate swing, a geochemical shift, a new developmental toolkit, and the system cascades into a new set of stable patterns? If so, the Cambrian Explosion is not a freak coincidence. It's one example of a deeper rule about how life climbs to new levels of organization, and that loops back to us. Because we are living inside one of these cascades. The neurons and body plans that first stabilized in the wake of the Cambrian are the scaffolding for everything we do. Our brains are elaborations of nervous systems that started as simple nets and jellyfish and ladder-like cords in worms. Our skeletons are variations on mineralized tissues that first appeared as tiny sclerets and shells on the Cambrian seafloor. Our eyes trace back to the first image-forming organs that helped Cambrian predators find their prey. We are walking, thinking consequences of a 500-million-year-old ecological arms race, and we're now doing something similar in culture and technology. A long build-up of knowledge, then sudden phases of rapid change that transform the landscape for everything else. So when you look at the Cambrian Explosion, 3 billion years of mostly microbial life, then a geologically brief blaze of animals, you're not just looking at a strange chapter in deep time. You're looking at a pattern that keeps repeating, in genomes, in ecosystems, in civilizations. Complexity often hides for a long time, then reveals itself quickly. The fossils and the molecular clocks together tell us that what looks like a sudden appearance is usually anything but. It's the visible crest of a wave that started far out at sea. And that's where the story leaves us. Not with a tidy answer to why every major animal group shows up when it does, but with a sharper sense of the question. Why does life so often move in fits and starts? Slow preparation, fast transformation. And what does that mean for a planet where one lineage from that Cambrian burst has learned to rewrite evolution's rules in real time? So when people say, for 3 billion years, nothing evolved, then every animal on Earth appeared at once, we can finally answer, that's not really what happened. For those first 3 billion years, evolution never stopped. It just worked mostly on things that didn't leave big, dramatic fossils. Microbes were evolving, metabolisms were evolving, entire planetary cycles were being rewritten by things too small to see. By the time animals show up in the record, the stage has already been under construction for an absurd amount of time. First, oxygen. Not a sudden gift from the sky, but a slow, glitchy rise. Microbes invent photosynthesis. Oxygen starts leaking into the oceans, then into the air. Iron rusts out of the seas. Toxicity spikes. Ecosystems crash and rebuild. Over hundreds of millions of years, that invisible gas creeps upward just far enough to make big, energy-hungry bodies possible. Then eukaryotes. Cells that swallowed other cells and turned them into tiny power plants. Cells with internal skeletons and flexible membranes. Cells that could stick together, talk to each other, specialize. The basic hardware for making tissues and organs is all eukaryote business. Then environmental chaos, snowball Earth events that may have frozen the planet nearly solid, glacial cycles, greenhouse rebounds, wild swings in nutrients and sea levels. Each crisis wipes the slate a little, reshapes coastlines, opens new spaces. Life that survives is life that can adapt fast. Then, the ecological spark. Once a few animals start moving, sensing, and eating each other, everything changes. Predators evolve. Prey responds with armor, burrows, spines, speed. Eyes get sharper, mouths get nastier. This arms race drives complexity far faster than a peaceful world ever would. Underneath all of that, the developmental toolkit. Hox genes and their cousins quietly wiring up front versus back, up versus down, segment versus limb. Tiny changes in when and where those genes fire can sculpt new body plans without reinventing biology from scratch. Once that toolkit is in place, evolution can remix it into trilobites, worms, early fish, and all the strange experiments of the Cambrian seas. And finally, the thing that makes it all look sudden to us: preservation. For most of Earth's history, even if animals had existed we might never have known. Soft, squishy bodies don't fossilize well. Shallow, storm-swept seas don't keep fine details. It's only when animals start growing hard parts, shells, plates, spicules, and when particular environments like Burgess-style mudslides or phosphatic seafloors lock them in place, that the fossil record turns on. So the Cambrian Explosion is what you get when all of that lines up at once. Oxygen high enough, eukaryote cells flexible enough, climate wild enough, ecological interactions intense enough, genetic toolkits powerful enough, rocks kind enough to actually save the evidence. From our point of view, compressed into a textbook timeline, it looks like a jump cut. Nothing, nothing, nothing. Trilobites. In reality, it's deep time doing what deep time always does. Take changes so slow you'd never notice them if you were there. Let them stack up over hundreds of millions of years. Then, when a few key thresholds are crossed, they cascade into something that looks, from the outside, like a revolution. Animals did not appear out of nowhere. They are what happens when a planet spends billions of years slowly re-engineering itself, chemically, biologically, geologically, until complexity stops being fragile and starts being inevitable. And we are one tiny, late-arriving branch of that story. Our nerves, our bones, our senses are all elaborations of tools first hammered together in those ancient seas. This sudden Cambrian bloom is still echoing every time a fish swims, a beetle crawls, a bird flies, or you take a breath. If you enjoyed this journey through deep time, you'll probably like where we go next. So please tap like, drop a comment, and subscribe so you don't miss future episodes. And in the comments tell me, where in the world are you listening from? And what time is it for you right now? If you want to dive even deeper into the strangest corners of prehistory, I've also put together a book all about hidden prehistoric secrets and the stories the rocks are still whispering to us. You can find it at sleepytimehistory.com. Because the more we learn about how long it took for animals, and for us to arrive, the more miraculous it feels that we're here at all, with the time and the curiosity to look back and ask how it began.